Good day to you, wherever you are in the world. Welcome to Cranfield University. My name is Toby Thompson, the Studio Director in the glorious Grenville Turner Studios here at Cranfield University. Thank you for taking the time out, for joining us. And if you're watching the recording, a special welcome. EBIC and Cranfield want to continue the conversation, so stay in touch. We're here talking about enzyme engineering for PFAS remediation. If you understood that title, then you deserve to be here. If you're into enzyme engineering, then welcome. If you're into PFAS and PFAS remediation, also welcome. But if you're new to all of that topic, then stick around, because coming up in the next hour, we're going to be together for no more than 1 hour. We're going to be talking about that with a range of experts. Now, we want your interactions. We want your questions, your comments, your challenges, pretty much at any time that we're together. If you can find, through the miracle of Microsoft Teams, the chat section, the chat panel, not the Q&A, just the chat, then type us preferably a complicated question or a deep technical question about enzyme engineering, because the people that we have here joining us in a second are hopefully going to be able to answer those questions. So feel free to put something in the chat now as to where you are in the world. We're always keen to hear where you are in the world. And we've also got at the very end some information about upcoming events in September, the conference in Southampton, and the next webinar event taking place on the 29th of July. But more on that later on. Joining me in the studio, Tao, looking good. You're looking very smart.
Thank you very much.
Are you ready for answering really complicated questions about enzyme engineering?
I'm kind of ready. If not, I will pass it to my colleagues, Dhiman and Natassja.
Well, I'm going to introduce Tao a bit later on, but we have Tao joining us, who's a Cranfield faculty here, and green technology and bioremediation by the sound of it, and wastewater treatment too. If that's your area, Tao is the person to ask questions of. Yes, we also are joined by greatness. Dhiman, give us a wave just to prove to us that you are real, not AI generated. It's fantastic. It's great to have you with us. Dhiman is joining us too. And Dhiman has a very interesting angle. Dhiman is looking at sensing and measuring. So if you're looking at either of those two things, sensing, how do you sense stuff? How do you measure stuff? Dhiman is the person to talk to. And we are in the presence of greatness. Natassja, give us a wave too. We were worried because you wandered off a few seconds ago. But we're with Dr Bush here, who's Chief Executive at Inspiralis and has a deep science take on everything that we're talking about. So stick around, because we've got more from Dhiman and Natassja coming. Can I start with you at first? Let's situate this. Let's just do a little bit of an embarrassing introduction to you first of all. Your work comes at the confluence, as I understand it, of environmental engineering, chemistry, nanotechnology. Have I got that right?
Yes, exactly. So my research area mainly focuses on using different technology for water and wastewater treatment.
Okay.
Yeah, and then mainly situate in bio-based and nature-based solutions.
So you've got a theme too, in terms of EBIC, a theme too, bioremediation. That's your angle on this whole thing. So you're interested in PFAS remediation, I'm assuming.
Exactly. So I work with the EBIC and work with other colleagues, and specifically focusing on the enzyme engineering, and also help with the other engineering biology approach for environmental remediation. Yeah.
So we're talking about PFAS remediation. Maybe situate the whole of this conversation, first of all, with what are we talking about in terms of PFAS?
Yeah, PFAS, I suppose all the audience, if you subscribe this this talk, you must have heard about the PFAS. So think about the chemical structures. So you probably think about different atoms, the carbon, hydrogen, oxygen, right? So PFAS is basically based on the carbon and the fluorine bond. So all the chemicals, if they have the carbon and the fluorine bond, it can be considered as a PFAS compounds. And it's been very useful. It's been firstly invented in 1930s. So around 80 years ago. And very fastly, there's some very famous product, for example, the Teflon.
Yes, we've all heard of that.
Yes,
we all use it.
How can we survive without it in the old time, but not now?
Oh, old. So
yes.
Teflon is old now.
Not old. I mean, it's because for the environmental researchers, we find out there's some toxic effect of the PFAS. So yes, it's very useful. But after so many years of usage, we find out there's a lot of residues in the water, in the sediment, in the soil, and also in our human body, different organs. So that caused the toxic effect to our health. So that's why we come to this technology innovation, try to develop the enzyme engineering technology for the PFAS remediation.
Remediation, solve, lessen, disambiguate. Say more about remediation.
Yeah, remediation. So for the conventional technology, they based on the chemical or physical process, for example, absorption. So that is absorb PFAS. You don't really break down the chemicals. So it's just a transmit from one place to another place, right? But for this approach, what we are trying to do, we are trying to use the biological process to completely break down the chemicals. So carbon, fluorine bond, we break it down. So it's not PFAS anymore. And then, yeah, totally remove it.
If you've not had a chance to understand where you can ask a question, you can ask a question pretty much at any time. So if anything we're not saying is clear, please put something in the text chat and we'll go over it. Just briefly then, why is it the most pressing challenge that we're facing in terms of chemicals, PFAS?
So PFAS, there's actually quite a lot of challenges in terms of removal. So firstly, of course, it's being considered as a forever chemicals. So forever chemicals means it stays there. Yeah. It don't go anywhere. It won't be break down. So it's the carbon fluorine bond is very strong. So it's very hard to break down, and so that's why a lot of conventional process they based on the either physical process to translocate it, to absorb it, or they use the chemical process to use the high energy demand process to break it down. But through this 1 hour talk, hopefully can give us some confidence and also some evidence to show we can actually develop the technology that can break down PFAS with a limited cost level.
You make it sound so simple. Confidence, evidence, inspiration. We want to inspire you that there's something to be had here, especially around enzyme engineering. To that point, this is a segue. Natassja, this is a first call to you. Natassja, you're actually working in this field. You're the Chief Executive Officer of Inspiralis. Tell us a little bit more about your experience of PFAS and what enzyme engineering is. And I know you've got a PowerPoint you want to share with us in a second.
Sure. So I have to admit, I'm not an expert in PFAS at all. Our relationship with Cranfield has introduced us to this forever chemical and it being a massive environmental challenge. Our expertise lie in enzyme engineering. So taking enzymes that are natural and then working with, changing their structure or their sequence to make them either more effective or more stable, or to hopefully make them break down things that weren't naturally take on as substrates. So although we don't have the strong PFAS background, we definitely have the expertise in the enzyme engineering side of things.
And you're chief executive, and you're also a scientist. You come from a scientific background. What do you wear each day? Are you chief executive or are you a scientist?
Oh, I'm hopefully somewhere in between, because you can't have one without the other when you're running a biotech company. Because the biggest thing is thinking about how we can leverage the biology and the science to make it commercially viable. And I think that's one area that engineering biology really needs to work on, is to make it a commercially viable product, is to think about ways we can scale it and leverage it and make it commercially viable.
You've got some slides. Tell us more about Inspiralis and enzyme engineering.
Sure. So yes, just thank you for having me on your webinar today. It's great to be able to sit alongside Dhiman and Tao. So as Tao's mentioned, PFAS remediation is a major environmental challenge, and there's a large market opportunity for those who can develop a lower impact, effective route to PFAS destruction. Just to give you a sense, the PFAS remediation market was valued at about $3.5 billion in 2024. And overall, the PFAS cleanup cost is thought to be about $200 billion in the US alone over the next 25 years. But this leads us to the question, as Tao highlighted, can we engineer highly selective and scalable enzymes for PFAS degradation? AI-assisted enzyme engineering is attractive because it may shorten the path from candidate enzyme identification to experimentally testable variants. The critical industrial question, as far as I'm concerned, is can AI design something useful, and can we validate, scale, manufacture, and deploy it? Slide, please. So who's Inspiralis? Well, we're a UK biotech company. We specialize in enzymes used in drug discovery, molecular biology, and advanced therapy manufacturing. The company has long-standing expertise in enzyme design, production, characterization, and supply. And one of our core strengths is translating enzyme biology into reliable products for scientists and industrial users. We are expanding now from therapeutic and molecular biology applications into environmental biotech. PFAS work is part of that strategic expansion. We're applying existing enzyme and manufacturing capability to this new environmental challenge. And to do this, we will need to use enzyme engineering. As I'm sure all of you know, there is no natural enzymes that degrade PFAS molecules. Next slide, please. So traditional enzyme engineering usually combines literature mining, so going out and finding what else is out there in the literature, whether it's an enzyme that naturally does something or does something near to this. For example, with the PFAS enzymes, it will be halogenases or those kinds of enzymes. Then looking for sequence homology, understanding the sequence, the structure of the proteins, identifying areas within the protein that you want to change, so mutagenesis, and then screening all the variants that are of interest. This approach is extremely effective, but it can be slow and it can be experimentally expensive. So when you look at it at just a few amino acids, the design space is enormous. Even a small number of amino acid changes creates a very large library of possible variants. So for example, if you had a protein motif of about 20 amino acids in an enzyme that you wanted to mutate systematically with only the aromatic amino acids, of which there are 5, that would give you about 2 million variants that you could potentially screen. So historically, the bottleneck here has been deciding which variant should we make and screen. So AI can help us prioritize better candidates earlier, reducing the experimental cost and time. And it does this through leveraging protein language models. Next slide, please. Protein language models are AI systems adapted from natural language processing, such as those used by GPTs, like ChatGPT. These protein language models treat amino acid sequences a little bit like language. So sequences contain patterns, they contain grammar words and phrases in the forms of motifs and domains, and they have context. And in our case, that's biological function. By learning from large protein data sets, these models can identify relationships between sequence and structure and sometimes function. And depending on the model, they can be trained from, there are various models out there at the moment, designed by a variety of people. Some of them are publicly available, some of them are paid for. So you've got, some of them have been trained on about 8 million to over 100 billion unique protein sequences. And that provides quite a lot of power. But at the basic level, they can help us answer questions such as which residues are likely important, which mutations are likely tolerated, which regions may affect stability or activity, and which candidate sequences are worth testing. So when we think about that, if you think about a protein, there are certain, under our understanding of structural biology, if you change certain amino acids, you will change certain structures within a protein. For example, if you have an alpha helice, if you put a leucine in place of a proline, you're likely to break the helix. So it's thinking about those kinds of changes and what those amino acids are going to do within the protein. But with the learning of over 100 billion unique protein sequences, that gives power that probably no single scientist will be able to do on their own. So more advanced tools are moving beyond predictive in generative design, so including de novo protein design, which may be a way of solving a PFAS problem. They're also able of doing biomolecular interaction prediction, such as those that are used in identifying antibody epitope interactions. Public examples such as AlphaFold 3 and afterfusion show how quickly the AI-enabled structural biology and protein design are advancing, and these tools are evolving, and new tools are being developed at a rapid rate. I feel like I open my computer every day and I see a new model that's being developed. But however, AI can't do everything yet. Next slide, please. So, what does AI change? AI can help with accelerating hypothesis generation. It can prioritize experimental work. It can combine information from public databases, structural predictions, literature, and proprietary data sets all together in a usable fashion. It can suggest mutations, flag likely stability issues, help rank candidates. But AI predictions are not products. Enzymes still need to be cloned. They need to be expressed, purified, assayed, stress tested, formulated, and manufactured. For environmental applications, they also need to be validated against real-world matrices, and the degradation byproducts need to be evaluated. And this also has to be in line with current and future regulations. If we broke a PFAS molecule down, you don't want it to be more toxic than the original molecule. So all of that has to be evaluated. And this is why the industry opportunity lies in connecting the AI design with robust wet lab and manufacturing and testing capabilities. With the potential power of AI to speed up the design of new variants, we've decided to build an AI enzyme engineering tool. However, we wanted it to do it responsibly and scalably. Next slide, please. So building an AI enzyme engineering tool is not just a software project. It requires biologists, protein engineers, data scientists, and software developers to all work together. We started off by using a low code or no code, also known as vibe coding, kind of prototyping early on, because it helped us demonstrate to the software engineers and data scientists exactly what our requirements were. And this quickly bridged the gap between scientists and software engineers and significantly reduced the cost of building bespoke software. One big challenge is that robust back-end development requires technically competent partners. And this is to ensure the correct architecture is put in place to protect partner and company data and avoid development corners that limit future expansion. And this is one reason why Inspiralis is working with Pelago, who are software engineers, to support development of a scalable and maintainable system, because we do not have those expertise. And the strategic value in that is not simply the tool itself. It is the in-house capability developed around the tool, leading to better data discipline, better experimental design, and faster learning cycles. So it's built as an entire workflow. AI and science are constantly changing. And this is why you need a development partner who will keep your tool relevant and tailored to business needs. And often when we're looking off the shelf or online tools, we felt were very generic, but we can leverage our data and all the data that's available to make something really powerful. And AI is arguably now part of daily life and business. Soon it will no longer be a differentiator for companies, but it will become a qualifier. Next slide, please. So the design that we went with is not too dissimilar to what you would do in traditional enzyme engineering. You still have to, all AI is doing is speeding up the process. So we work on a design, build, test, learn cycle. We define target activity and performance requirements. So if you're looking to increase activity, so you would look at cofactor binding, you'd look at substrate binding, you'd look at various, on-off rates. And from there, we gathered relevant public and private data, and we're leveraging that information to do things like structural searches, hotspots identification, mutagenesis, structural validation, and then scoring and ranking. And then these come out with optimized enzyme variants or de novo enzymes. And then these selected candidates move into wet lab testing. And once we have gone through that process, all the experimental results, positive and negative, are fed back into the system to improve the next design cycle. Thinking particularly about PFAS, Cranfield's role in this is quite critical. On the data side, they understand the PFAS chemistry, the degradation pathways, and they have expertise in molecular modeling and dynamic simulations, which we don't have in-house. On the wet lab side, they have expertise in analytical validation and environmental testing, all of which inform the AI system what it should be optimizing for. Next slide, please. So as I think you might have gathered from that, data is the real differentiator, and model choice matters, but not quite as much as high quality data matters. Public data sets are extremely valuable. They've got a collective knowledge within them on sequence structure, enzyme annotations, literature, and known activity data. And the bigger data sets, so the subscription and curated data sets, can add further depth to that information, but private data sets are often where the competitive advantage emerges. This includes internal data sets, assay data, failed variants, which are not often reported in the literature, expression data, and then moving forward to purification performance and manufacturability. In environmental enzyme engineering, partner data, as I've mentioned, is critical. For example, being able to leverage the molecular docking and dynamics data, including understanding the pollutant chemistry and the degradation products, give us kind of real sample conditions to be able to feed into the system to continuously improve the variance that the system ranks. The most valuable AI systems improve because experimental data is captured cleanly and reused. However, when you're using both private and proprietary data, data governance becomes very important. Next slide, please. So we're focused very heavily on responsible implementation of building AI tools safely. So AI tools in biotechnology need to be responsibly implemented from the start. DevOps and database architecture matter and need to be robust. A useful AI system must be maintainable, version controlled, and integrated into real scientific workflows. We always make sure that we use enterprise versions of models to keep confidential prompt sequences and outputs to make sure that they're not used to train public systems. And we always try and make sure that at every step within our system, we're using the correct model, because the correct data is what's going to give you the best outcomes. So yeah, there are different tools that are better for structure prediction or sequence generation or stability assessments. Something which is critical in enzyme engineering is biosecurity. So we need to control for this, and the systems that we're building should avoid generating or optimizing dangerous biological outputs. And this will be through flagging or just not allowing those kinds of things to happen. Cybersecurity is obviously very important. Access controls, audit trails are essential because sequence data, customer data, and assay data can all be commercially sensitive. And I think the biggest thing for us is that the system is never finished. As science models and data sets evolved, the tool has to evolve too. And just as AI systems kind of need to be scalable, I'm going to switch gears a little bit now and talk more about another challenge in scaling, and that is scaling engineering biology. Next slide, please. So as I mentioned, scaling is a common challenge in engineering biology. A promising enzyme is only useful if it can be expressed, purified, stabilized, and manufactured economically. There are a few key scale-up questions that need to be considered. Does the enzyme express well in a production host, well enough that it can be easily purified? Is it soluble and stable? Can it be purified efficiently? Does it retain activity under process conditions? Can it be formulated for storage and deployment? Is it compatible with closed loop treatment systems or stable long enough and active in real world conditions? For example, with wastewater treatment, a lot of enzymes work exceptionally well at 37 degrees Celsius or 52 degrees Celsius for some molecular biology enzymes. But when you put them into wastewater, they're likely going to need to be active at 10 degrees. So we need to make sure that they're active and stable in real world conditions. And does its deployment clash with current regulation? And this is part of this is where Inspiralis's enzyme production experience is useful. And it also where we're going to be, be able to work with Cranfield in leveraging their expertise and their their knowledge, which is makes this a very powerful partnership. The future of AI enzyme engineering will depend on collaboration and connecting design tools to manufacturability data as well. Next slide, please.
Natassja, any chance I can just stop you there for a second, because we are running out of time, but there's a couple of questions, if I can put those questions to you. We may have time to come back to the slides that you have just coming up. But Maria, you asked a question that I was keen to ask on, and Natassja, you kind of touched upon it. Frankenstein compounds, aren't we at risk of creating Frankenstein compounds resulting from remediation of PFAS?
Absolutely. I mean, there's always the chance that when you break something down, that you make it worse than what you started with. But I think that's something that has to be built into your design of your enzyme. And it has to be something that has to be tested systematically to make sure that you're breaking it down to something that can be either broken down by something else that's available, or it can be broken down further that it is no longer toxic. But obviously those toxic intermediates have to be broken down extremely quickly. And understanding the chemistry of the molecule is very important in that. And that's something that we have to prioritize when we're designing variants.
But you sound pretty confident that you're not going to create something worse than what you're trying to solve.
Well, I would say that is a fundamental criteria that has to be met. So you're not going to release an enzyme or produce an enzyme. It will kill the project immediately. There's no point in having something that can do a first step, create something more toxic, and then you've created something that's not useful. It's part of making a solution that does what it says on the tin. Otherwise, it's not economically viable as far as we're concerned.
Brilliant. Maria, we're keen to hear more. If you want to come back, please do that. One more question, if I can, Natassja. This is from Avray. Avray is talking about deployment. How do you deploy enzymes for contaminated soils, for instance, or water? How do you go about that?
So there are a couple of different ways that you could do it. I think when you're looking at a lot of the environmental bioremediation, sometimes enzymes might not be the best option. You might look at engineered microbes or something like that, where you've got long-term bioremediation. Enzymes, I think, are going to be most effective in short-term bioremediation. So, for example, inline wastewater treatment, where you attach it to a substrate in a bioreactor, and the water passes through that and is remediated in that before it comes out clean on the other side. It could be used in a closed loop system. So as Tao mentioned earlier, you extract PFAS from the environment. There are physical extraction methods such as using activated carbon or things like that, but that PFAS needs to be degraded. So what you could do in those situations is a bit like adding washing powders, the best thing I can think of right now. So a dried enzyme to that to then break that down, which it can then be safely disposed of, as opposed to needing to be incinerated or something like that.
Natassja, thank you.
Can
I just add? I think Natassja has covered all the aspects. Just a little bit added on. So in terms of the deployment, so we worked together with Inspiralis. So for example, that's just one example. Is we try to immobilize the enzyme. So the enzyme, if you spray it directly, so it's a kind of free movement. It's mobile phases. But if we can coat the enzyme onto some solids or some particles, so to immobilize that, and then it can become a filled bed reactor, and then it can become a reactor, and then the water passing through it, the reaction can happen. So we will also make sure there's no modified enzyme or other toxic compound come out of the reactors.
That's good to hear. Thank you. If I can move on to Tao, you're next, but somewhere in your presentation there was a question, a follow-up question from Avray about where is the biggest source of PFAS pollution? Don't feel you have to answer that right now, but you've got some slides for us. Tell us a bit more about your area of research.
Yeah, of course. So nowadays, so basically PFAS is everywhere, and there's a contaminated soil, the water, and even air. And then if you follow some recent research publication, they find the PFAS in the very remote desert area, mountains, and also our human body, different organs. So very serious risks. So that's why it comes back to the topic, to find a way to do the appropriate remediation is very important. Yeah. So I prepared some slides, and I think just to follow up Natassja's presentation, we are currently collaborate. And Natassja, Inspiralis, they are focusing on the enzyme engineering, and we are focusing on very chemistry. So we have very chemistry expertise, focusing on the PFAS degradation, and also we have some modeling experience. So it's a good match to find to develop the technology innovation. So here, if you look at these slides, these two compounds, the PFAS, one is a P4A, one is a PFOS. Those are the two typical PFAS, and those are the first two PFAS be regulated in the US. And it's still two most typical PFAS being always concerned in the environment. So here you can look at the structure, so carbon and fluorine, and you can see there's a lot of fluoride, right? So the chain of a PFAS can be longer, can be shorter. So this is to give you an idea about how the PFAS looks like. And I put two numbers at the bottom.
What are those?
Yeah. So the left side one is, you see, here are two examples of PFAS. How many PFAS species in the world? So if you look at the US EPA definition, or the EU, there's an institute called the EU Classification and Labeling Institute. You can find the number of PFAS is between 4,000 to 7,000 PFAS. And the right side, 7 million.
Incredible.
It's in the academic area. So how we define the PFAS. So as long as the chemical structure, they have a carbon and a fluorine bond, and we can consider it as one type of PFAS. So there are 7 million of them. So loads different PFAS species in the world, in the environment as well.
A bigger challenge.
Very big challenge, and different chain lengths, different structure, their property, the chemistry property are different. So that caused a lot of challenge to the application, to the remediation.
Incredible.
Yeah. Next slide, please. And here I put the legalization at the top, but please note that some of them are not actually legal requirements. For example, in the UK, probably you have heard of the DEFRA PFAS plan. That is a non-bind guidance. But here you can see in different countries, the requirement of the PFAS specifically in drinking water, they are different. So for example, in the US, they required in the drinking water, each single PFAS, the concentration should be below 4 nanogram per liter. And in EU, they look at 20 PFAS species. The cumulative level should be below 100 nanogram per liter. And if you look at all PFAS, it's 500 nanogram per liter. But in the UK, it's also different. So the DWI, so it's the directive for the drinking water requirement, and then we look at 48 PFAS species. If you think about how many PFAS species in the environment in the world, we focus on 48 of them, that most concerned ones. And then the requirement is each single of them should be below 10 nanogram per liter, and the total of them should be below 100 nanogram per liter. So here give an idea about the requirement of the concentration level. Just to give you an idea, so nanogram per liter is a very, very low level of concentration. But still, if it's beyond 4 or beyond 10 nanogram per liter, still will cause the toxic effect. So really, PFAS cause quite a lot of concerns in terms of, so we really need to find the technology for the remediation. Next slide, please. Okay, so this picture was taken in the US in Princeton University. So last year, I fortunately got the support from EBIC. I traveled to a research partner from Princeton University. And if you look at the different color of this bottle, so Toby, can you do your estimation about why they have different colors and what's this for?
I have no idea, but it looks very pretty. I know it's not pretty.
It is pretty, and the function is also prettier. So basically, they got the specific bacteria, they cultivated the bacteria in the lab, and then this bacteria can actually break down PFAS.
Ah.
So they demonstrate that.
So that orange one has broken some elements of PFAS down.
No, the orange one is the suitable environment, the specific bacterial strains they can survive.
Ah.
The transparent one is the control.
Gosh.
So the bacterial strain grows there, they can actually break down PFAS. So that brings, this is one of the first research group in the world that they find out some naturally occurred bacterial strains, they can actually biodegrade PFAS. Yeah. Another question, and also for the audience as well. So can you imagine where they got these bacterial strains?
Where it came from?
Yes.
Naturally occurring, by the sound of it, not. Sounds human-made.
Sounds human-made.
Okay.
Yeah. Let's look at the next slide. So it's from the nature wetland.
Oh, wow.
Yeah, so this is Professor Peter Jaffa from Princeton University with his postdoc and a student. So they collect the soil sample from the nature wetland in north part of New Jersey, and then they bring the soil back, and then they do the cultivation. They find some specific bacteria.
And that's bad, I'm assuming.
Good.
That's good. Why
Yeah.
is it good?
So firstly, we demonstrate, because before, you remember we talked about the conventional PFAS removal technology, physical, chemical, right? Incineration, burn it. That's cost effective, not cost effective. And but if we find the nature occurred bacteria, they can break down the PFAS.
That makes sense.
So we can use that, right, to break down the PFAS. So
Naturally occurring.
yes. So that is the evidence, the direct evidence to show PFAS can be biodegraded. That can be biodegraded. Yeah.
Now we're talking.
Down. Yes.
That's good news.
It's good news. And through this, so this is nature occurred bacteria. But what we are trying to do is say, how can we accelerate the process, right? But not working with the bacteria itself. Right. So if we work with bacteria, it's a little bit complicated. If we do the gene editing, and then it's kind of with synthetic new bacteria, and then they have the horizontal gene transfer. They may transfer the gene to other bacteria, and then they may grow. So if something happens, it releases to the environment, that causes the problem, right? At least currently the policy haven't. Have the right policy to say whether we can release the gene modified bacteria or not. So the policy part is one of the development, but we are working with the technology. So instead of doing the gene editing of the bacteria, we're trying to do the modification of the enzyme. So enzyme is different. It's a substance released by the bacteria. But actually, all the functions of break down the chemicals is produced by the enzyme, right? So can you please move to the next slide? So this is a work we have done. So it's sponsored by both the EBNet, one of the UK Research Council sponsored network, and also within EBIC. What we have done is, if you look at the first graph in the background, so that blue and light green color, so that is the enzyme. It's a protein structure. So we managed to get the 3D protein structure. And then if you look at the chemical part, those are the PFAS. And one is a short-chain PFAS, another is a long-chain PFAS. So through this work, we are doing the molecular docking and the molecular dynamic simulation. So we, based on the 3D structure and the energy, the enzyme can produce to help with the break down the chemical structure, and also calculate the carbon-fluorine bond to say whether this energy can be higher than the energy needed to break down the chemicals. So we do the molecular docking and molecular simulation. We can say where the PFAS compounds can locate it, in which location of the protein, and how much energy needed to break down the PFAS. So through this modeling, we can help to provide some information to the AI derived modeling, the enzyme design. So that is Natassja mentioned. So AI has been used for enzyme design. That's very powerful, but they need a lot of information to train the data. So through this modeling, we can say which enzyme or which part of the enzyme we can do the modification that can help with the breakdown the PFAS. And then we can provide a lot of information to the AI model and then train the AI. And then that can help us give more information to do the enzyme engineering modification that can accelerate the process of PFAS degradation. Yeah. Next slide, please. So this is the last slide. We through EBIC, we have a different collaboration. So this is a photo I took last week. So me and the left side is Professor Paul Jeffrey and Dr. Yasmin Mida.
I recognize Paul.
Yeah, both of them are working in EBIC as well. And Paul is working on the policy part and the technology governance. We had a visit to Japan, a different institute. And then if you look at this one, is one of the National Institute for the Engineering Biology. If you look at the background of the machine, of the instrument, it's called, it's part of the biofoundry. The biofoundry means that's, you remember we do the modeling, right? And then we using AI to design the enzyme. What's next? So we design, we need to build, and then do the testing, and then and have the learning, and then go the round and the round. So it's not just the modeling part. We need this physical infrastructure to help with the enzyme construction and then do the testing. So this is part of the instrument in Japan, but also I want to use this. So this is all automate. So if we do the manual process of building up the enzyme, that costs a lot of time. But this is automated. Everything is automated, and that can accelerate the process of testing the technology. So although this is in Japan, but also bring back to the collaboration why we think it's very important and necessary to work with Natassja, with the industry. So through their platform, we not only have the AI modeling platform, we can also use this high-tech instrumentation to help with the whole testing process that can support the future deployment, as we discussed.
It's a very positive picture you're painting, which is refreshing to hear. Thank you. Put your questions to us if you can. There's a couple of questions, one from Mohammed and one from Avra again. I'll go possibly to Mohammed's first, and then possibly to go to Dhiman's session. But is the fluoride released by the PFAS breaking enzymes more dangerous for dehydrated people in, let's say, Qatar or Middle East suffering the heat? Is heat a factor there?
It's one of the possibility, but if it's a free fluoride, so normally it's, but compared with the PFAS compound itself, free fluoride is less toxic. But before the discharging, we normally would like to harvest the free fluoride as well. So I remember, you remember I put a photo of the partner from the Princeton University. Yes. So last year we had a PFAS workshop. And we had we invited to speak from the Oxford University. You know what they are doing? Very interesting. So they use not the biological process, use the physical process to break down the PFAS, and they harvest the free fluoride, and then make it to the biomedicine. So they complete the circle of reuse. Yeah, to break down the pollutants and reuse the different chemicals. So I think that is one of the possible way we can go for it as well.
Wow.
Yeah.
More on that story in a second. We want to come on to Dhiman if we can in a second. Please keep your questions coming. We are very keen to hear from you. Dhiman, let's wrap this up and put your angle on what it is that we're talking about here. Which angle are you coming at this conversation from? And you've got some slides for us as well.
So first of all, I would like to thank the organizer for giving me the opportunity to appear in this podcast. So my job has become quite easier because PFAS has been introduced by its length and breadth by Tao and Natassja. So there is another angle that I want to bring to this discussion. So every management starts with detection first. Unless you can detect the compound, unless you can monitor the compound in an easy way, you cannot come up with effective mitigation strategies. So can I have my slides, please? So in the interest of time, I'll just go through the slides. So to the next slide. Yeah. So so PFAS has already been introduced, but I just want to mention that PFAS is as Tao mentioned, it's a large family of synthetic chemicals that have been extensively used in industrial processes. And but unfortunately, this same stability thing has become a problem because of its high stability. It is become a persistent environmental contaminant, and as Tao said, PFAS accumulates in water bodies, soils, even in human body, and it is raising a significant concern regarding its environmental and public health. But coming to the PFAS management, it's a multifaceted approach that requires parallel deployment of conventional approaches, then policy interventions, public awareness, in fact, use of PFAS alternatives as well. So we at EBIC, we want to use engineering biology for PFAS management. So with engineering biology, we can expand as Tao said, we can expand the natural ability of the microbes to degrade PFAS more efficiently. As Natassja mentioned, we can take out the enzymes, modify it using AI touch, and then make the enzyme even more suitable for PFAS degradation. And of course, engineering biology approaches are eco-friendly and often cost-effective. Having said that, all management starts with the PFAS detection. So as Natassja already touched upon, so if we just have a peek into the PFAS testing market, you can see it's a several billion-dollar global water testing market, and within that market, PFAS is a growing niche. You can see the graph here. The North America is the biggest market, followed by the European market, and Asia Pacific, China and India are also coming up with the regulatory limits, and this regulatory limits is one of the key driver of this healthy growth rate of PFAS testing market. So this is a regulatory limit map of the Europe. So you can see, as Tao already mentioned, the permissible limits are quite low. Those are in PPT level. PPT is nanogram per liter. So we need to detect the PFAS to make the process efficient. To make the PFAS management process effective, we need to detect PFAS at PPT level. So that is the main restraint of this market. So if we just have a look at the current detection method. So currently the LCMS method is the gold standard, but LCMS is a costly affair, although very effective. But its per sample costs around $200 to $300 and it has high turnover time, it requires centralized instrumentation, skilled manpower, so it increases the cost. So if we see the scale, if we see the contamination and the regulatory compliances in terms of testing, so we are not matching it up. So if we can have sensors which can cost effectively detect PFAS in a short time in a field deployable manner, so we can cut down the cost, and the money saved can be diverted to increase the coverage. So that's why we have to have sensors. Now coming to the sensor, can you go to the next slide? Yeah, coming to the sensor, so there is a debate whether should we have a chemical sensor or biological sensor. So essentially, sensor fundamentally consists of two components. One is the recognition element, which I call the receptor, and a signal transduction mechanism. So the traditional chemical sensors often rely on receptors like polymers, ionophores, nanomaterial for analyte recognition. While the biosensors, this recognition element is a biological entity. It may be a antibody, it may be an aptamer or peptides or a protein that can effectively bind to PFAS molecule. So the key advantage of biosensor is that they are highly specific, and they are able to distinguish closely between related compounds. So this makes biosensors particularly attractive for PFAS detection for where high selectivity is very essential. Now coming to the challenges. So as Tao already mentioned, so the regulatory limits are in picomolar or femtomolar level, and so we need to make a sensor that can go to that low level of detection. And then stability, often the biologically recognition elements like proteins or enzymes are not stable, so that is a main issue. And then affordability. So to address these challenges, a lot of private and public bodies are working on that to develop cost-effective PFAS sensor. Can you go to the next slide? So aptamer is one of the bioreceptor that we are working on. So these are short nucleic acid molecules capable of binding specific target compounds with high affinity and specificity. And as Tao already mentioned, that PFAS is not a single compound; it's a group of compounds. So and some PFAS molecules are structurally quite similar. So you have to have specific detection. So aptamers are customizable. We can screen aptamers for structurally similar compounds as well. So recent studies have identified aptamers capable of recognizing important PFAS molecule like PFOA and PFOS. These two are the model PFAS compound, as Tao already mentioned. So we took these aptamer sequences, and we developed an assay by which we showed that yes, these aptamers specifically interacts with PFOA. So for that, what we did a very simple assay. We just tagged the aptamer with a fluorophore and blocked that fluorophore with a quencher molecule, which is attached to the attached to another nucleic acid, which is base paired to the aptamer, so that the quencher and the fluorophore comes together. So in presence of PFOA interacts with the aptamer, and the quencher molecule gets separated. So we see increase in fluorescence. So it was good, but the thing is the jump in fluorescence was not that great. So we have to have a signal amplification. So to address that limitation, we developed a strategy that couples aptamer recognition with rolling circle amplification. So the concept is very straightforward. So in absence of PFOA, the primer remains blocked within the aptamer structure, and when PFOA binds to the aptamer, the primer is released. And the released primer can initiate something called rolling circle amplification, which basically it's a plug-in machinery by which a DNA molecule is amplified with the help of DNA polymerase enzyme. And this process generates a large amount of amplified DNA product from a single recognition event. So the amplified DNA is then easily detected by a detection module if you attach a detection module to that. So since the DNA molecule is already amplified, there are many molecules of DNA, so they give a much stronger fluorescence signal. So in essence, the biological recognition event is converted into a nucleic acid amplification reaction, providing substantial signal enhancement. Can I have the next slide? Okay, so here you can see in the graph that I have shown. So if you don't attach the signal amplification module, we get some signal, but if we attach the signal amplification module, the signal is amplified more than 10-fold. So that's how we are amplifying the signal so that we can easily detect by simple fluorimeter. So with this method, we could detect PFOA to a concentration range of 0 to 40 ppb, which is a big range considering the PFOA's natural limits. And also we could achieve a limit of detection of to the PPT level. And this result demonstrates the potential of combining synthetic biology inspired amplification systems with molecular recognition elements to create a sensitive PFAS biosensors. Now, can you go to the next slide? So we are there, but still we are not here. So to improve the sensitivity and specificity even further, so we are trying to introduce a second bioreceptor in the form of peptides. Now the peptides are very attractive bioreceptor. They are highly specific, very stable, and they are customizable. So they also can be used as a PFAS capture element. So for that, we got a grant from EBIC, which is called as EBIC Flexible Fund. So with that grant, we'll be screening peptide molecules, small peptide molecules using fast display library. It's a standard method of screening short PFAS binding peptides, and those peptides will be used to capture PFAS, and then we'll club it with the aptamer based recognition so that we can have a hybrid strategy. In this design, PFAS molecule will be first captured using the immobilized peptide-based receptors, and subsequently the aptamer primer construct and DNA amplification systems will be used to generate highly amplified signal. So this combines the strength of both the recognition systems. The peptide component act as a capture element, while the aptamer and DNA amplification module provides exceptional sensitivity. Okay, so such dual recognition system may ultimately improve both sensitivity and specificity for real-world environmental samples. So to conclude, I must say that PFAS contamination represents a global environmental challenge that requires new monitoring technologies, and our work is mainly focused on combining engineering biology, molecular recognition, and nucleic acid amplification to develop a rapid and highly sensitive biosensor for PFAS detection. And we have already demonstrated aptamer-based detection of PFAS with rolling circle amplification for enhancing the signal. And we are now extending the platform through peptide discovery using fast display library screening. Ultimately, our goal is to develop affordable, field deployable sensing technologies that can support large-scale environmental monitoring and complement the conventional laboratory methods. So thank you for your attention. So I'll stop here.
Dhiman, thank you so much. If you can sense it and measure it, I guess you can manage it. So again, another very promising input to the conversation. Thank you so much. Please keep your questions coming and stay in touch with EBIC throughout all of your interest, certainly beyond the time that we're together. We have a few more minutes. I want to come back, if I can, very quickly to Natassja. You mentioned scaling earlier on. There's a question here about scaling. How do you scale enzyme engineering in a very short amount of time that we have?
Well, there are two different streams. You've got to think about your upstream and your downstream. Once you have your variant that's of interest, you need to consider how much enzyme you're getting out per liter of fermentation. We're very lucky with molecular biology enzymes because you get a lot of enzyme for your expression. It's extremely active. So we're getting, microgram, high activity per microgram, so high specific activity. Sometimes with environmental enzymes, this is not the case. So understanding that first parameter is really important. And that leads into your downstream. So we can get away with molecular biology enzymes of 30-liter fermentations, and you get billions and billions of units of enzyme. With environmental applications, you have to consider the size of fermentation you need to get the equivalent enzyme activity out. So sometimes that can be 1,000 liters of fermentation, which is an entirely different scale. So you have to start small and then increase your scale that way. So, but it's also having the infrastructure to do thousands of liters of fermentation, which there are very few in the UK that can do that. We're busy working on our next scale plans. We haven't quite got to the 1,000 liters yet, but we're looking at whether or not we outsource that or do it in-house. And then with that comes the scaling of your downstream process to get highly active pure protein out, because you don't want other contaminants there that might affect the activity and the way the enzyme works in closed loop systems. So it's thinking about the best way, quickest way, and shortest way you can extract the enzyme from the organism that's producing it.
Natassja, thank you so much, and we're very happy that Inspiralis is working on this same thing. Tao, can I come to you very briefly? We have about less than a minute remaining. Concluding thoughts.
All right, so that is a very heavy task. So I think the conclusion thought after this, I won't say let's not just stop after this webinar. So this is a very fascinating research area and also deployment, because that directly links to our human health and the public health. So just want to let everybody know. So we have a bunch of people. We working on this area, try to find the most cost-effective and efficient way, which is enzyme engineering for the biodegradation of PFAS, working together from the academic and the industry. And also we have some policy colleagues all worked together. So we are open for all the collaboration and looking forward, be positive and to achieve the best, the sustainable best environment for us to live.
Stay in touch, I think I heard there. Stay in touch with EBIC and finding out what's going on. We have run out of time. It just leaves us to say that there is the conference that I mentioned earlier on, September 16th to 17th of this year in Southampton, the Biology for Environmental Solutions. There's an early bird, so get yourself booked on that. We also have another one of these events taking place on the 29th of July online. So please look out for both of those. Natassja, thank you so much indeed for spending time with us today, whichever hat you have on today, scientist or chief executive. Dhiman, it's been fantastic for the deep dive that you've done, so thank you. Sorry we couldn't spend more time with you. And Tao, wow, you the man. So thank you for inspiring all of us.
Thanks for having me. Yes, thank you.
Thank you for joining us today. Stay in touch with EBIC and we'll see you next time on the webinar. Thank you very much indeed.